Good afternoon. Today is Sunday, May 31st, 2020. My name is Ricardo Sandoval, and this video is for week 3 of Biology 225, Microbiology at Los Angeles Pacific University for the Summer 1 term. And this week I'll be talking about Lab 4, which is titled Microscopy for Microbiology. So I'll be going through the anatomy, if you will, of my microscope, and then we'll also be looking at some slides, different magnifications, and things of that sort. For this part of the experiment, there isn't any specimens, so to speak, that require any sort of PPE. So with that said, I'm going to go ahead and then flip the camera and then start with the anatomy of the microscope. All right, so anatomy of a stethoscope. So this is my eyepiece here. This is what I look into, basically down this way, all the way here, so I can see what's going on. There is a slight magnification here, as you can see, of 15 times, and I've noticed there's a little kind of pointer in there that just kind of helps identify things when I'm looking into them. So this is the eyepiece tube. This is what's called the coarse adjustment. So this I use basically to locate whatever I have on a slide. So this makes the, I guess, body, if you will, of the stethoscope go up and down, so I can get closer to or farther away from a specimen. So these here, this is the coarse adjustment or the fine adjustment. So once I've achieved my desired heights, I can go back and forth, of course, forward. It makes it a bit finer, and back kind of just adjusts that. So this here, this piece here, this is called the arm of the stethoscope. Here and here I have stage clips. These just hold the slide in place. This, of course, is the base. This is called the main tube of the stethoscope. This piece here is called the nose piece. And these here are my objective lenses. So they turn and lock into place, and the lens, of course, that's in the back, is the active lens. So this small green lens is my low power lens, and it is a 4-time magnification. This blue lens is my medium, and it is 10 times magnification. And then this red lens here is my high power magnification, and it is 40 times magnification. And then I also have here, this is called an oil immersion lens. This is 100 times magnification. And how this works is I'll have a slide here, and it comes with this little flask of oil, and then just one drop of oil goes on top of the specimen, and then this lens is used to, I guess, further see things here. So that's this part, objective lens. This part here is called the stage. Under here, this is called the disc diaphragm. And then there's a mirror right here. And then, of course, this is the lens. And then this is a light box that I turn on so that I can see what's going on. All right, and that's the anatomy of a stethoscope. So for the remainder of this first part of the lab, I will be viewing prepared slides. So there won't be much for me to show you what I'm doing, but I will upload photographs of these prepared slides at the optimal magnification level into my laboratory report. Okay. I also did want to point these two knobs here, and these are just stage adjusters, very useful when it comes to adjusting a slide. And as you can see, this knob here adjusts a slide left and right, and this knob adjusts the stage or the slide itself up and down. These two knobs are very useful because even though it appears that when you place a slide on the stage that it's properly aligned. A lot of times, depending on the objective lens being used, I did have to adjust the left and right and up and down position of the slide in order to view the specimen in hand. All right, so for this next exercise or part of the lab, I'm going to collect my own specimens. So one's going to come from the inside of my cheek, one's going to come from my teeth. So we're looking for cheek cells, and I'm also looking for basically plaque, from my teeth. So I did want to go over the supplies that I have for this part of the experiment. So I'm going to flip the camera. And then as far as PP that's required, are my goggles, my gloves. I've already wiped down my surface with my 10% bleach solution. Okay, so as you can see, I have two slide covers. I have a pipette, and then I have these cotton swabs, one that I'm going to use to collect the sample for cheek cells, and then one that I'm going to use to collect the plaque. I, of course, have my microscope, and then in here, I have a bit of distilled water that came from this wash bottle. So what I'm going to do, and I'll do my best to show this to you, is I'm going to take a cotton swab, rub it on the inside of my cheek. I will then spread that sample on the slide, put 1 drop of distilled water using this pipette onto the slide, and then cover it with the slide cover and then view it under the microscope. Same thing for the plaque. I will grab the other swab it pretty firmly against my teeth, place the swab on the slide, just drop of distilled water using the pipettes, slide cover on the slide, and then under the microscope. I will take pictures along the way. So with that being said, I'm going to go ahead and get started. All right, so I'm going to place my pipettes into my distilled water here just to make it easier access. And then as these swabs are sterile, I'm just going to push one out of the packaging just to kind of maintain their sterility here. And again, I'm going to start with my cheek sample. So I, of course, won't be able to talk during this part. Okay, so that's a pretty decent sample. So I have my slide here. So I'm just going to smear this all over the center of the slide, just making sure that I do turn the swab itself completely around just to make sure that I get the maximum cells. All right, so there's that. I'm gonna throw this in the trash. Okay, and then now using my pipette, I'm just gonna drop a tiny bit of my distilled water. I don't need very much here. And then just 1 drop's gonna go down. Okay, this then makes it a wet mount slide. My slide cover, and then just gently place the slide cover on the edge of the water and down. Now there is a tiny bit of excess water here, so I have a paper towel that I'm just going to dab with, and as you can see, the paper towel is absorbing the water quite easily, because I don't want excess water here, because it'll make viewing my cheek cells that much harder, especially since the cells themselves are not stained. There's no color to make them more easily viewable. Okay, so that's my cheek cells. I'm just going to put them to the side here so I don't mix them with the next specimen that I'm going to collect, which is going to be my plaque cell. Plaque cells from my teeth. So my last swab, this is going to go in the trash, and this time I'm going to place the Q-tip or the swab against my teeth near the gum line to get a sample. Okay, so there's that. And then again, I have my sample here. My slide here. I'm just going to rub it using the entire slide, the entire swab here all over the slide, just to make sure that all the cells are visible and applied to this slide. Okay, there's that. Again, this is going in the trash. And then again, 1 drop of distilled water. Okay. And then my slide cover goes on top of that. Okay, and then again, I'm just going to use my paper towel because there is a tad bit of excess water again. Okay, so plaque and cheek. So at this point, my next step is to take some photographs of these as is and then place them under the microscope for the optimal power. Steps that are really kind of hard to talk about as I'm doing via the microscope, but I will upload photographs of these and my completed report.